Okay, so if every cell why isn't it recording? Hold on. Oh, it is. Oh, it is. Now that's going to be in the recording. That's cool. Hi, recording. DNA to RNA to protein. If every cell needs DNA, therefore, we need a way to make sure that we can replicate it faithfully, right? If the proteins function, then we don't want a done goof, because if the DNA is screwed up, then the RNA is going to be screwed up, and the protein is going to be screwed up. So we need a foolproof way of replicating DNA. So let's remember really quick about the structure, because you haven't seen this 74 times. Just kidding, you have. When we're looking at the nucleotides, remember, phosphate group, 5-carbon sugar, nitrogenous base. If you don't have that yet, get that. Get that in your brain hole, because it's super important, okay? Remember that when we're looking at the different nucleotides, they have nitrogenous bases, and those are going to be either pyrimidines or purines. Always one bound to the other. Okay, we're going to look at the discovery of DNA. We're going to look at these scientists, Griffith, Avery McCarty, and McLeod. I don't know if it's McLeod or McLeod. I feel like it can't be McLeod, but I've always heard it as McLeod. Do you know? I'm calling it Mac. Calling it Avery and Mac. Hershey and Chase, Chargaff, Wilkins and Franklin, and Watson and Crick. Lots of groups. So let's go ahead and start with Griffith. Okay? I have dates on here. That's just to give you an idea of how this went sequentially. This is not in what year did Griffith? No, I'm not AP US history. So we're thinking late 1920s, and here's the experiment. 2 strains of bacteria, one pathogenic, one non-pathogenic. And when you heat treated the pathogenic bacteria, it kills it, right? So if you heat treat the bacteria and you put it in a mouse, that little guy lives, even if it's the pathogenic strain. But if you heat treat the pathogenic bacteria, mix it together with non-pathogenic bacteria, somehow that bacteria that was non-pathogenic before got something from the dead bacteria that allowed it to become pathogenic again. So there was some type of heritable information that the otherwise non-pathogenic bacteria picked up, and it was able to kill this poor little dude right here. Rip. Okay, so the conclusion made from this is that the living non-pathogenic bacteria somehow transformed, that is a key word here, because this is a transformation experiment, into the pathogenic bacteria. But of course, at the time, Griffith didn't know what exactly that was. Avery, McCarty, McLeod came around, and they just repeated the same thing. Now, this is 15 some odd years later. Repeated the experiment, but this time, they purified out either the proteins or the nucleic acids. When they purified out the proteins, the experiment still worked. When they took out the nucleic acids, the experiment didn't work. So that meant that it must have been the nucleic acids that were in play here. Do we see how this is starting to develop, right? We're getting closer to figuring out why or how heritable information is passed along. Okay, here's the problem. Most people were very, very I don't believe you. You've done something wrong. Because it doesn't make sense that a chemical with only 4 bases could have ever possibly been responsible for making us, making our diversity. We must need more complexity in our genetic material, because if it's just DNA, that's only 4 letters. There's no way that's possible. So Hershey and Chase wanted to add to the evidence to support that nucleic acid was the genetic material, or not necessarily that they wanted to show that, but they wanted to see for themselves. So they took radioactive sulfur, radioactive phosphorus. What do we know about sulfur? Where are you going to find it? In proteins. Where do you find phosphorus? Right? So if you take radioactive sulfur, then only the proteins are going to become radioactive. If you take radioactive phosphorus, then only the DNA is going to become radioactive. And they put it into a bacteriophage, which is just exactly what it sounds like. What is phage or PHAG mean? To eat, and bacterio means bacteria. It's a virus that infects bacteria. Okay? So these bacteriophages were labeled with either radioactive sulfur or radioactive phosphorus. And when the phosphorus was used, the bacteria became radioactive. Ergo, therefore, whatever. It must be the DNA, it must be the nucleic acids that are in the bacteriophage that allowed for the genetic information to be like passed in. Ergo. Look, I even wrote ergo. Questions so far? Okay, so we're mounting support. Chargaff comes along, 1950s. He sees every organism that he tests has slightly different amounts of adenine and thymine versus cytosine and guanine. But in everything he tested, those two were pretty close. The amount of A always was statistically close enough to the amount of T to call them the same. Same can be said for C and G. He didn't know why, but he was like, neato. Okay? Wilkins and Franklin. I want to talk to you about something here in a sec, too. So, Wilkins and Franklin, it was Wilkins' lab, it was Franklin's work. That's what you get for being a grad student. You give someone else the credit, so technically Wilkins got the credit for this, but that's okay. They used what is called X-ray crystallography. So they basically took a beam of X-rays, shot it at crystallized DNA, and it bounced all over the place. And it made this pattern right here. And that pattern is indicative of a molecule that's helical in nature. So they were like, whoa, it's helical. Or it forms like a spinny thing. Okay? So now the evidence is there for how to make a model of DNA, but no one had done it. Finally, oh, this is wrong. Not 1928, 1953. I'm gonna put that in quotes because it's like, that's what someone said, but it totally wasn't me. In the 1950s, Watson and Crick are now going to take the experimental evidence, and at this point, scientists are still on the protein train. The scientists at the time were like, stop studying nucleic acid. It's not the genetic material. It's useless. It's stupid. And they were like, okay, But they still wanted to figure out what the structure of it was. So they used the information from the other scientists, Chargaff, Rosalind Franklin, to be able to build this model. And after a lot of failed attempts, they finally figured out that the nitrogenous bases face inward with a purine and a pyrimidine always bound together. This was huge. In doing this, they also realized that the structure of DNA can give a hint as to how it's replicated, which is why I wanted to go through these scientists. At the very end of Watson and Crick's 1953 publication, I'm pretty sure that's right, they made reference to the fact that it has not eluded them that there is a mechanism of replication inherent to the structure of the DNA. We'll talk about that in a second. Okay? All right, I want to take a second and tell you about your term project, because one of my favorite books is all about this. Over the course of the year, I mentioned earlier that you will have some type of project. Most people do it at some point in the fourth marking period, but if you want to get a head start on it, you may. One thing you may do is read about anything biologically related. But Watson and Crick actually published a book about their discovery, and if you want to do like a little read it, present on it kind of thingamabobber, that can count as your like year-long project. So remember, your final is made up of 3 grades, your midterm, your final, and this project. That can be an idea for a project if you want to do that. Remember, if you take the AP exam, then the best 2 out of 3 scores is what goes in for your final grade. Okay. I just wanted to mention that real quick. Moving on. Okay, let's talk about replication. Go over a quick overview, go over the specifics, think in your mind, structure, and function. Okay, so Watson and Crick were the first to suggest that the structure of DNA could explain how it replicated, right? The fact that C and G always go together means that when you separate it, it can both act as a template for the new synthesis of more DNA. That's brilliant. That's so elegant. That's a wonderful realization. Okay? Mieselson and Stahl, make sure you're up here. Okay. Mieselson and Stahl did an experiment that gave credibility to what Watson and Crick had proposed at the forefront, which was they took heavy nitrogen and where are we? We're over here. I should probably point so people watching at home, what up? Okay, sorry. So when you take the heavy nitrogen, what you would expect is that if this conservative model held true, which means that after DNA replication, there's one new strand and one old strand. If that were to hold true, then you should have equal amounts of heavy nitrogen and light nitrogen, or heavy DNA and light DNA. And that wasn't the case. So we knew it couldn't be the conservative model. In the dispersive model, chunks of old and chunks of new ended up in each new strand. And so you would expect that all the DNA would be like halfway between heavy and light. And that was factual. But then, when you replicate it again, you would expect that that becomes slightly lighter, right? Because you're using now light DNA nucleotides to build the new strands. So the new strands should be light, or the newly synthesized portions should be light. And when you mix it with the old stuff, it should make an average that's somewhere all the same. Eden, you're looking like, what? No. Okay. When Mieselson and Stahl did this experiment, the first round, they did find that the weight was somewhere in between the heavy DNA and the light DNA. But after a second round of replication, there was about half of it that was heavy and light, kind of mixed, and then about half of it was light, meaning that it was completely newly synthesized strands of DNA. So that's giving credibility to what is known as the semi-conservative model. Okay? In other words, partly conservative, conserved. The DNA is partly conserved in each replication. And if you can't really see it, that means if these are the original strands, then that's going to be used to make new strands. And then in the next replication cycle, there's a total of 4 strands here. 1, 2, 3, 4. One of those is still going to be completely original, and 3 of those are going to be new. The other strand of DNA that was originally here is going to have the same thing. 1 old strand and 1, 2, 3 new strands. Okay, so the Mieselson and Stahl experiment demonstrated that DNA replicates via a semi-conservative manner. So what is that semi-conservative manner? Well, let's start by taking a look at the DNA of a prokaryote. It's a big old circle, okay? And there's a location called the origin of replication. And that origin of replication is going to be the origin of replication. Wow! Meaning that is where replication is going to start. So in this case, it's drawn in black. We're going to have helicases. Helicases are enzymes that separate out the DNA, and they're going to separate the DNA, move outward, so that now you have this bubble. And this bubble is full of single-stranded DNA that can now act as the template for new replication. That makes replication forks at each end, kind of that Y-shaped area, if you ever hear me talk about a replication fork. Okay, so far so good? Easy peasy lemon squeezy? Okay, so you have now been introduced to the helicases. Why are they my favorite enzyme? Helicases Unzip your jeans. Get it? I'm special. Okay, so now we're at the replication fork. Right, we're right here. Here's our helicase. It's coming through and it's separating. It's breaking those hydrogen bonds between the A's and T's and C's and G's. Helicase, chugga, chugga, chugga. Now, have you ever taken yarn and separated it out? What happens when you pull yarn apart? What happens just upstream of where you've pulled it apart? It gets like super tightly wound, right? If that's your DNA, that's gonna break. And that's not good. So we have a topoisomerase, which is this guy right here, whose job is just to kind of break the DNA in a controlled way, allow it to spin, and fix it. That's the job of the topoisomerase. It's just relieving tension so that the DNA doesn't completely break apart, because you can't put DNA under as much tension as you can yarn. Also, as you're separating the two strands, we've got these little guys here, single-stranded binding proteins, and their entire job is to bind to the single strand of DNA. And that's just protecting them from reclosing back on themselves. It's kind of like a little Ziploc. This is just like the spacing between the two Ziplocs that ensure that it doesn't re-zip on itself. Okay? So how do you separate out the strands? Sure, you need helicase, but you also need other players involved, topoisomerase and the single-stranded binding proteins, to ensure that this entire complex remains stable. Now, when it comes to actually replicating the DNA, primases are going to be needed. And so the primases are going to add what are known as RNA primers. They're just adding little short segments of RNA that is complementary to the DNA. Why? Because DNA polymerase, which is what's going to be adding the new nucleotides, DNA polymerase cannot bind to single-stranded DNA. It just doesn't fit. The substrate, which is the DNA, doesn't fit into the active site. So you need two strands here. One strand is DNA, the other strand is a primer of RNA. So DNA polymerase 3 is going to bind, start adding in the 5' to 3' direction, and it's just going to chug along right behind the helicase. So here's the helicase. It's been moving out of the way. Then the primase, that's been moving in, or sorry, not the primase, the polymerase moving in, following along. Everything is good. Easy? Questions? Okay, you guys forgot something. Everything's good so far, right? I've got my 5' to 3' being built. But DNA runs antiparallel, which means if this strand, the top strand that's being copied, is being read from 3 to 5, we can't just go this way towards the replication fork on the other strand. Oof. So what are we going to do? DNA polymerase 3 can follow the helicase only in one direction. On the other strand, it's going to go in the opposite direction. So follow with me here. Take a look up top. If this is your origin of replication, originally the two dark blue strands would have been one strand. That got pulled apart. And in one direction, we've been able to build 5' to 3'. Let's go ahead and number that. If this is 5, you can't see that. If this is 5 to 3, then this DNA must be 5 to 3'. So far, so good? Okay. And if that's the case, then this is 3 to 5 here, which means I can still, from my origin, build out in the 5' to 3' direction this way. So that's okay. Those are called the leading strands. And the leading strand is happy. It's just chugging along. Helicase is opening up the replication fork in this direction, opening it up in this direction, and at each end, you have one strand being synthesized well.
Question.
Yes.
I think the way you number them doesn't make sense.
Oh yeah, that says 3. I don't know what the problem is. Why are you saying there's a problem? There's no problem. Did I fix it?
Yeah, they all were like looking around.
1 to 3, 1 to, yeah, now it's right. Okay. So on the leading strand, everything's Gucci. On the lagging strand, That means that primers are being built right where the helicase is, and then replication happens away from the replication fork. So replication is going to happen that way, separation is going to happen that way, and now we're going to have a new segment of DNA that we need to add on to. So that means we need to have, why are there little black boxes there? That's annoying. What are known as Okazaki fragments, which are those little segments of DNA that happen on the lagging strand. So let's follow through at what happened. At one point, the replication fork was right about here where my little red dot is. A new primer was added, polymerase built in the 5' to 3' direction, and helicase continued on this way. So then a new primer got added, DNA polymerase added in the 5' to 3' direction, and the helicase continued onward. New primer, new building, new primer, new building. Follow? Okay? So that leaves us with all these little fragmented pieces with RNA stuck in between. So for starters, once we get to the end of a segment, the DNA polymerase 3, DNA pol 3, is going to fall off when it hits the next primer. DNA pol 1, right here, is going to be what is replacing the primers. Okay? So it's taking the RNA out and replacing them with DNA. Then we just need a phosphodiester bond in the backbone of the DNA between a sugar of one and the phosphate of another, and that's going to be the role of the ligase. It's just ligating or sealing up that last tiny bit. Okay? So the leading strand has one primer, one DNA polymerase 3, it just goes. The other strand, it's going to make these little segments of Okazaki fragments and then have to connect them. Okay, I also want to mention quickly about repair. Repair is going to be really important, and repair is done by DNA polymerases, not necessarily 1 and 3. There's others out there, but you don't need to know which ones. But basically what's happening is as the DNA is being paired up, if there's a mistake, DNA polymerase can proofread it. So it can double check that if there's an A here, there's a T there. If there's a C there, there's a G there. If this doesn't work, which 1 in a million times it doesn't, that's when you lead to a mutation. If you have a mutated DNA, then the RNA is going to be different. Then the protein is potentially going to be different. Okay. So that's all I had to say about all of that.